This is the second set of notes for the um, biochemistry unit. Although technically the third set of slides that you've seen, the first one again was general chemistry. You just should have watched a video on water molecules. This is talking about carbon, and there will be one more video to follow. When we talk about carbon, carbon is basically the backbone of life. Living organisms consist mostly of carbon-based compounds besides water. It is able to form huge, complex, diverse molecules, and we will talk about why. Proteins, DNA, carbs, and lipids, they are all composed of carbon compounds. So there's 4 large macromolecules. Carbon is so important that it has its own study, organic chemistry. This is why in college, if you take Gen Chem, you'll end up eventually taking organic chemistry and biochemistry if you are pursuing any sort of medical field. Most organic compounds contain hydrogen as well as oxygen, nitrogen, and phosphorus, but they are usually really long chains of carbon with some hydrogens thrown in. To talk about why this is so important, if you remember the Miller-Urey experiment, the Miller-Urey experiment talks about how we went from non-living carbon to living molecules, or the synthesis of organic compounds. Essentially, we know that methane gas, ammonia, hydrogen gas, and water were all present in the atmosphere when the Earth began post-Big Bang. We know that when we sealed them inside glass tubes, and we basically used electricity to synthesize lightning, we saw that we started making organic carbon molecules, which is the basis for cells. And then we went through the endosymbiosis hypothesis, how one cell engulfed another cell to make our eukaryotic cells. That's neither here nor there. The important part is that this Miller-Urey experiment allowed us to determine how we make carbon molecules, which is where life begins, truly. Again, this goes over it. You have seen this experiment before, so this shouldn't look super unfamiliar, but pause here if you need to and take a look at it. The reason that carbon is so capable of making these huge macromolecules is that carbon has 4 valence electrons. What that means is that unlike a element with 8, which is not going to bond with anything, and an element with 1, which needs 8 or 7 valence electrons, a carbon molecule has 4 valence electrons already and therefore can make 4 bonds. It can form really large complex molecules because it can have another element or another atom bonding on each side. A single carbon molecule can make a tetrahedral shape, shown on the right, but if we string long chains of carbon together, they can create these huge macromolecules. Typically, we see carbon, nitrogen, oxygen, phosphorus, and sulfur bonding together. This is because they all have 4 or 5 valence electrons, therefore allowing them to make a similar amount of bonds. When 2 carbon atoms are joined by a double bond, the atoms joined are on the same plane. This is important because shape of a molecule dictates its function, and so when we have this long straight chain, it's going to have a very specific function, and we'll get to that a little bit later. You do not need to memorize the shape of the different carbon molecules, but if we look at this, all of these are very linear shaped molecules, whether there's a single bond between the carbons or a double bond, and this has a purpose, which we will talk about. Here are some examples of more carbon skeletons. Carbon chain can vary in length. They can vary in if they have single or double bonds. They can be branching, as shown below. They can also create rings, which we see in your nucleic acids. DNA and RNA are deoxyribose and ribonucleic acids. Those are going to be ring structures that are very stable carbon circles. Hydrocarbons are a very specific category of carbon molecules consisting of only carbon and hydrogen. Most organic molecules, such as fats, have hydrocarbon components, and they undergo reactions that release a huge amount of energy. This is why when we talk about carbohydrates and eating foods that contain these hydrocarbon molecules, they are going to release a lot of energy when their bonds are broken, because we talked about how as bonds break, they are releasing energy, and that is where the energy from those carbohydrates is going to go towards your mitochondria and be processed through cellular respiration. When we look at fats, fat is going to be another hydrocarbon structure, also very highly energy containing, but obviously it is stored in different ways. When we talk about these carbon molecules, we need to understand the term isomers. Isomers are the same molecular formula but different structures. And if you recall back, I talked about how the shape of a molecule dictates its function. And so I can have C6H12O6, right? 6 carbons, 12 hydrogens, 6 oxygens. But they can arrange themselves in a bunch of different ways depending on what its purpose is. There are 3 types. You don't need to know it in very much detail, but just know that structural isomers have different covalent arrangements, so different bonding, same structure, but like maybe a double bond somewhere else. Cis-trans isomers have the same covalent bonding, but different actual like visual arrangements. And then the last set of isomers are enantiomers, and they are isomers that have mirror images. They're just rotated slightly. Here's a structural isomer. A cis-trans isomer, we see that they are just kind of flipped. And then we see that these are the mirror image isomers. But again, these isomers you don't need to know. The really cool thing that might help you remember this, though, is that the last type, we have the mirror image isomers, are super important in the pharmaceutical industry. Basically, they a mirror image drug of a carbon molecule is going to have a very different effect. Usually one isomer is biologically active, and then when we flip it, it has a very different purpose. When we look here, we see that ibuprofen, there is an effective and an ineffective isomer. Albuterol, what is in your inhalers, again, there is one structure that is effective for treating this and one that is totally ineffective. And so our body is very capable of recognizing which of these it is ingesting and whether or not it's going to be useful or not. And so again, this goes back to the idea that the simple structure of these carbon molecules dictates what its function is and whether or not it is functional at all. When we get to proteins specifically, we talked about functional groups or R groups. A few chemical groups are key to determining the function of molecules. The carbon skeleton is usually very basic, and then a number of groups attach a hydrogen, and we call them functional groups or R groups. Functional groups are components of this organic molecule that are usually involved in the reaction itself, and they have very unique properties. So if we take a look at this, so we see that on the bottom on the example, we have ethanol, but its functional group or its R group on the far right, where a hydrogen would normally bond, is going to be your hydroxyl group. It has very specific functional properties. It's polar, and it can form hydrogen bonds with water molecules, dissolving compounds such as sugars and things like that. So if I were to want to have a very specific purpose for ethanol, I would attach a hydroxyl functional group to it. Carboxyl, C double bonded with O. This is a ketone and an aldehyde. It basically, it has to do with processing sugars, and has to do with, like, when we talk about diabetes and stuff like that. We'll come back to this. Carbonyl, and then carboxyl is going to be COOH, similar as well. Amino, NH2, and so again, you can flip through these at your own pace, but these are examples of where normally we'd see a hydrogen attached. We have this functional group or this R group attached that has very specific purposes. And the AP test does expect you to know some of these. And so in class, we will cover which ones you are expected to know. A very important R group or functional group is going to be the phosphates and the adenosines that are part of ATP. We know that ATP is or adenosine triphosphate is a phosphate molecule, so not a carbon molecule, but still important. The adenosine is a compound, and then the phosphate groups, there are 3 of them, triphosphate, right? Tri 3, they attach to the adenosine. these triphosphate groups are broken off when we want to release energy to use that ATP. And technically, those phosphate groups are a functional group or an R group. What is important to know here is that the versatility of carbon makes it possible to have so many diverse organic molecules, and this is the foundation of where we get biological diversity.